Disaccharides consist of two monosaccharides joined through an oxygen bridge. In biological systems, monosaccharides exist in the cyclic form, and as we have seen, they are actually hemiacetals. Recall that when a hemiacetal reacts with an alcohol, the product is an acetal. In the case of disaccharides, the alcohol comes from a second monosaccharide. The acetals formed are given the general name of glycosides, and the carbon-oxygen bonds are called glycosidic bonds. And so you can see here that what's highlighted in yellow in alpha-D-glucose is the hemiacetal, and the hydroxyl in the hemiacetal is going to react with the hydroxyl on carbon 4 in this molecule of beta-D-glucose, and the atoms that are circled in pink, those are going to form the water molecule that is lost when we form this glycosidic bond. And the glycosidic bond is here highlighted in yellow. And it's important to point out that if you look at the anomeric carbon here, that anomeric carbon has an oxygen and an R group, another oxygen from the glycosidic bond to another R group, and then it also has a hydrogen and a carbon attached to it. So therefore, that is an acetal. And the reason we call this an alpha 1,4 glycosidic linkage is because the bond coming off of the chiral carbon is going down, right? It's pointing down, and remember, down is alpha. So alpha, and then the 1 comes from the anomeric carbon, and the 4 comes from the other carbon. So the bond is pointing down off of the anomeric carbon. That's the 1. Then we put a little arrow towards 4 because we formed the bond with the hydroxyl from carbon 4, and then of course alpha again because it's pointing down. Glycosidic bond formation is non-specific. That is, it can occur between a hemiacetal and any of the hydroxyl groups on a second monosaccharide, but in biological systems, we commonly see only particular disaccharides like maltose, shown down here, lactose, or sucrose being formed. These specific disaccharides are produced in cells because the reactions are catalyzed by enzymes. Each enzyme catalyzes the synthesis of one specific disaccharide, ensuring that one particular pair of hydroxyl groups on the reacting monosaccharides participates in the glycosidic bond formation. If alpha-D-glucose and a second glucose are linked, as shown down here, the disaccharide is maltose or malt sugar. This is one of the intermediates in the hydrolysis of starch. Because the carbon 1 hydroxyl of alpha-D-glucose is attached to the carbon 4 of another glucose molecule, the disaccharide is said to be linked by what? An alpha 1,4 glycosidic bond or glycosidic linkage. Maltose is a reducing sugar. Again, any disaccharide that has a hemiacetal hydroxyl group is a reducing sugar. This is because the cyclic structure can open back up at this position right here to form the free aldehyde. Disaccharides that do not contain a hemiacetal group at carbon 1 do not react with the Benedict's reagent and are called non-reducing sugars. What that means is if you were to take this hydrogen here, if you were to convert that into any kind of R group, then you would have an acetal here, and then you would convert this into a non-reducing disaccharide. Again, in order to be a reducing sugar, you've got to have a hemiacetal. Milk sugar, or lactose, is a disaccharide made up of one molecule of beta-D-galactose and one of either alpha or beta-D-glucose. Galactose differs from glucose only in the configuration of the hydroxyl group at carbon 4. In the cyclic form of glucose, the carbon 4 hydroxyl is pointing down, and in galactose, it's pointing up. In lactose, the carbon 1 hemiacetal hydroxyl group of beta-D-galactose, shown right here, is bonded to the carbon 4 alcohol hydroxyl group of either alpha or beta-D-galactose. It's beta-D-galactose that's shown here. The bond between the two monosaccharides is therefore a beta 1,4 glycosidic bond or linkage. Again, the reason this one is beta is because the bond going from the anomeric carbon is pointing up. So we would call this beta-D-lactose because this beta shown here, comes from the fact the hydroxyl at the anomeric carbon is pointing up. Lactose is the principal sugar in the milk of most mammals. To be used by the body as an energy source, lactose must be hydrolyzed to produce glucose and galactose. Note that this is simply the reverse of the reaction that we saw on the previous slide. Glucose liberated by the hydrolysis of lactose is used directly in the energy harvesting reactions of glycolysis. However, a series of reactions is necessary to convert galactose into a phosphorylated form of glucose that can be used in cellular metabolic reactions. In humans, the genetic disease galactosemia is caused by the absence of one or more of the enzymes needed for this conversion. A toxic compound formed from galactose accumulates in people who suffer from galactosemia. If the condition is not treated, galactosemia leads to severe mental disabilities, cataracts, and early death. However, the effects of this disease can be avoided entirely by providing galactosemic infants with a diet that does not contain galactose. Such a diet, of course, cannot contain lactose and therefore must contain no milk or milk products. Many adults and some children are unable to hydrolyze lactose because they do not make the enzyme lactase. This condition, which affects about 20% of the population of the United States, is known as lactose intolerance. Undigested lactose remains in the intestinal tract and causes cramping and diarrhea that eventually can lead to dehydration. Some of the lactose is metabolized by intestinal bacteria that release organic acids and carbon dioxide gas into the intestines, causing further discomfort. Lactose intolerance is unpleasant, but its effects can be avoided by a diet that excludes milk and milk products. Alternatively, the enzyme that hydrolyzes lactose is available in tablet form. When ingested with dairy products, it breaks down the lactose, preventing symptoms. Sucrose is also called table sugar, cane sugar, or beet sugar. Sucrose is an important carbohydrate in plants. It is water-soluble and can be easily transported through the circulatory system of the plant. It cannot be synthesized by animals. High concentrations of sucrose produce a high osmotic pressure, which inhibits the growth of microorganisms, so it is used as a preservative. Of course, it is also widely used as a sweetener. In fact, it is estimated that the average American consumes 150 to 170 pounds of sucrose each year. It has been suggested that sucrose in the diet is undesirable because it represents a source of empty calories, that is, it contains no vitamins or minerals. However, a recent study has determined that there is a significant relationship between excess sugar consumption and an increased risk for cardiovascular disease-related mortality. In addition to the link between sucrose in the diet and dental caries or cavities, has been scientifically verified. Sucrose is a disaccharide of alpha-D-glucose joined to beta-D-fructose. The glycosidic linkage between the alpha-D-fructose and beta-D-fructose is quite different from those that we have examined for lactose and maltose. This bond involves the anomeric carbons of both sugars. This bond is called an alpha-1 beta-2 glycosidic linkage since it involves the carbon-1 hemiacetal hydroxyl group on the anomeric carbon of glucose and the carbon-2 hemiacetal hydroxyl group on the anomeric carbon of fructose, noted in red. Because the alpha-1 beta-2 glycosidic bond joins both anomeric carbons, there is no hemiacetal in this molecule, sucrose. As a result, the ring can't open to the linear form, and sucrose will not react with the Benedict's reagent. Thus, sucrose is not a reducing sugar.